Welcome dear students. I am Richa Shakya, Assistant Professor from Noida Institute of Engineering Technology, Pharmacy Institute, Greater Noida. Today I am here to deliver the lecture on the subject that is a pathophysiology. The subject code is BP204T and this subject comes under the B Pharma and this B Pharma is a course offered by this Dr. APJ Abdul Kalam Technical University. Now, so today this is the last lecture of this unit. This is lecture number 29 and this is the last lecture of this unit. That is the topic of today's lecture is pathogenesis of cancer. In the previous lectures we discussed about the principle, the etiology and basically the classifications of the cancer. Now, at the end of this topic we are concluded the entire what is cancer, what is the classification of cancer and what is the etiology of cancer. At the end the pathophysiology of or pathogenesis of the cancer we discuss, or we at the end we are know about what is the entire cause of the cancer and what is the treatment for the cancer. Now, we discuss about the introductory part. So, in the previous lecture we already discussed about what is cancer. So, cancer is a disease which is basically caused by the abnormal or uncontrolled cell growth with a continuous manner, and when the cell continuously grow into the particular area and they forms the abnormal mass of the tissues, they called as the tumor, right. So, tumor is basically it is a non-cancerous type of cells, but when these cells are these are the benign, right. These are benign tumors and there is another thing is there is a malignant tumor. So, malignant tumors basically these are the cancerous cells, right. What is malignant? When the injured or when the infected cells are transferred from the primary origin to the secondary origin and now they get damage the secondary area, just like the person is suffering from the breast cancer, right, and after that these cells now migrated and transferred to the lungs, right. So, there is a malignant tumor or the metastasis cancer. So, basically benign tumors are the benign, and which means they do not invade or they do not migrate anywhere, means they stay on their original location. They do not migrate anywhere or they do not invade any other tissues other than the origin instead of their origin area, right. They cannot penetrate or they cannot invade to the other side of the tissues, they just stay on their original area, right. So, tumors may be benign and which means they do not invade nearby healthy tissues, but sometimes they are malignant, right. Malignant means when the origin cancers or the original infected cell migrated or invade to the another healthy tissues. So, these are called as a malignant, in which the genes are deviate, causing them to grow and divide erectly and spread into the nearby tissues. So, basically tumors benign and malignant. Always remember benign means stay at one place, but in malignant they just invade and migrate nearby cells or tissues. Right. So, always remember do two things: benign, which always stay their original location, and the malignant, which can migrate from their original location to the next one nearby, and they get infected and they just get damage to the nearby cells. As the cancer cells continue to grow, they may break off the original growth and travel throughout the blood or the lymph fluid to the other parts of the body. Genetic changes that can be inherited or the result of DNA damage caused by the external exposure like excessive ultraviolet lights, carcinogens like tobacco and smoking. So, basically cancer cells continues with the growth of many kind of things, just like person who are suffering, who are just continuously exposure with the sunlight, continuous exposure with the ultraviolet rays. So, due to this what happened, the person suffer from the skin cancer, right. And when the person is continuously intake of the alcohol, consumption of this tobacco, and they continuously doing the smoke, then person is suffer from the liver cancer or the lung cancer, or tobacco may cause the mouth cancer. So, there are the various kinds of cancers with the series of the events. Now, epigenesis or the epigenetics of the cancer. So, the classical view of cancer is a set of series that are driven by progressive genetic abnormality that include mutation in tumor suppressor genes. So, basically tumor suppressor genes and oncogenes and chromosomal abnormalities. Basically the classical view of cancer is a set of disease that may be derived from the various kinds of abnormality, various kind of abnormal series, which includes the mutation in the tumor suppression genes. Mutation in tumor suppression genes means the cells or the genes which may suppress the tumors. Suppress means just inhibit the growth of the cells, but what happened at that time, they just mutated. After the mutation they can't suppress the tumor suppressor genes, so they continuously grow abnormally and uncontrollably, right. This is the first point. Second is oncogenes or the chromosomal abnormalities. Chromosomal abnormalities means there is any genetic conditions where the chromosomes get infected, right. The genes, just like in the previous lecture I discussed, that is a BRCA1 and BRCA2. These types of cancer, these types of genes, when they mutated, what they do, they will cause breast cancer and ovarian cancer, right. There is a various genetics disorders, just like these are the two examples. So, epigenetic and alterations means epigenetic alteration are the functions which relevant with the modification of the genomes that do not changes the nucleotide sequences. So, what is epigenetic alterations? So, epigenetic alteration is just modify to the genomes and they do not modify the nucleotide sequences. The sequence not get altered, but what happened at that time, the modification in the genes, there is a modification in the genes, but not in the sequences. So, the example of such modifications are change in the DNA methylation, that is a hypermethylation and hypomethylation. Means there is a process of methylation where the methyl group will change. So, there is a changes in methylation, it may be hypermethylation or the hypomethylation. Histone modification, these histones are the proteins, ठीक है. So, histone modification and changes in the chromosomal architecture which cause the inappropriate expression of the proteins, just like HMGA2, HMGA1. So, these are the histone proteins which may changes their structure, and just like they shows expression just like HMGA2, HMGA1. Each of these alterations regulate gene expression without altering the DNA sequences. So, they get mutated, they get altered their functions or their gene expression, but without any DNA sequences. Means just like if the series is A, T, G, C, right, this is a series, or A, T, C, G, or A, T, T, U, G, there is a series, there is a various kind of series. So, there is a no change in the series, but there is a change. Where is a change? They change into the gene expressions, they changes into the gene expressions, but they can't change their sequences. So, that is why the cells can't recognize what happened at that particular area, because sequences is same. So, these changes may remain through the cell division, last for multiplication generation. It can be considered to be epimutation, which is equivalent to the mutation. Means when the DNA sequencing can't change, so the cells can't recognize the series, because the series is okay, but there is a generation, there is a changes in the gene expressions. So, this is known as the epigenetics, or this is just equivalent to the mutation into the genes. So, this is the flow chart, this is a flow diagram of the epigenetics, which discuss like endogenous agents and the endogenous agent which occurs inside the body, or exogenous which exposure from outside the body, right. So, endogenous agent just like diet related, just like a bile acids, macrophages and neutrophils produce ROS or RNS, ठीक है, reactive oxygens and the RNS, they are endogenous agents, they are just secrete inside the body, and they just mutated, the genes get mutated itself. Now, if you talk about the exogenous agents, so just like smoke, radiation, metal, viruses, other genotoxins which can comes inside the body through the any kind of exposure, right. So, what they do? They just do the DNA damage, they just damage the DNA, and what they happen when the DNA get damaged, the epigenetics alteration occurs, by which the DNA try to repair itself, right. But there are so epigenetic alteration just like miRNA, CpG, island methylation, histone proteins modification, chromatin remodeling, these are the agents which are help in the DNA repair, they are just act like a DNA repair genes. But just because of mutation in these kind of genes, the DNA can't repair itself, right. The DNA can't repair itself, so DNA get damaged continuously. So, if the DNA can't repair itself, the DNA repair deficiency, because DNA can't repair. Now, due to this large increase in unpaired DNA damage, so there is a pairing, there is a pairing of, just like this is a DNA, right. So, there is a pairing. So, the large increase in unpaired, so there are some series which are unpaired, so they get damaged at that particularly area, right. Now, large increase in the somatic mutation and the epigenetic alterations, they what they do, they just multiply, alter the field defect, the drivers of the mutation, and which is the initial progression of the cancer, right. Side by side there is a epigenetics or somatic mutations with the many primary non DNA repair genes which cause the somatic mutations and the epigenetic alterations, and these somatic and the epigenetic alterations cause the progression of the cancer. So, these are the various kind of series which occurs or which cause the cancers. So, basically I conclude this slide in the simple way that there is a exposure of the any kind of substance from the outside or from the inside the body, that is the exogenous or the endogenous agents. Endogenous agents just like a bile juices or macrophages, or there is some just like neutrophils production, that when the neutrophil production is very high, then there is a reactive oxygen is very high, there is a inflammation occurs. So, this is a condition inside the body which means endogenous agents will cause. Now, exogenous agents means just like dietary products, just like microbes, environmental conditions. So, these are the exposure agents. They what they do, they just damage the DNA, they just damage the DNA. So, there is a some kind of DNA repair genes, right, there are some kind of DNA repair genes, but these agents damage these DNA repair genes. So, when the DNA repair genes get damaged, so what happened at that time? At that time DNA will not able to repair itself. So, DNA get continuously damaged. So, the series of the damaged gene is continuously, because in the previous lecture in the previous slide I told you that there is a mutation in the genes, but they can't mutation in the series of the DNA. So, there is a no changes in the DNA sequencing, but there is a changes in the genes. So, when the DNA repair genes get damaged, so there is a DNA sequencing, there is a DNA sequencing is unpaired. So, these unpaired DNA sequences continuously, they goes continuously, and after that they cause the DNA damage, and after that due to this there is a continuously cell growth, cell division, and ultimately they cause the progression of the cancer. Now, pathophysiology of the cancer. So, I already told you there is exogenous and the endogenous. So, acquired, just like acquired and environmental, just like DNA damage agents, just like chemicals, radiation, virus, these are endogenous, sorry, these are exogenous. Right. So, these exogenous agents, when there is a normal cell, they react to the normal cell, and that what happened at that time, DNA get damaged, but there is a no problem, there is after that DNA normal when the normal cells get damaged, they perform their activity on the normal cells, the DNA get damaged, but DNA repair itself successfully. Now, in some cases just like inherited, like endogenous, like DNA affecting DNA repair, genes affecting cells growth and apoptosis. These are the conditions which occurs inside the body. So, when there is an alteration in the inherited mutation in the gene affecting the DNA repair, gene repair. So, there is a DNA gene repairs, when they get damaged. So, what happened at that time? the DNA can't repair itself. They can't able to repair itself because the DNA gene is already damaged, right. So, at that time there is a failure of the DNA repair. So, when there is a failure of the DNA repair, so there is a mutation in the genome of somatic cells. So, when the mutation in the genome of the somatic cells, there are various series, just like first is activation of growth promoting oncogenes, second alteration of genes that regulate the apoptosis process, and third is the inactivation of cancer suppressor genes. So, first is they activate the promoting cells of oncogenes. They just provoke, they just activate the oncogenes. Now, second one is they alter the process of apoptosis. They can't know how they do, how do they get apoptosis, how to program cell death. They can't know. So, they do not know. So, there is an alteration of the genes that regulate the apoptosis, and there is an activation of the cancer inactivation. There is an inactivation of the cancer suppressor cells. When there is a cancer or tumor suppressor cells get depressed or they are inactive. So, what happened at that time? There is a regulatory control cancer cell will develop. Now, then these series, now expression of altered genes products and loss of regulatory genes products, these cause the series of functions just like clonal expansions, addition of mutations, additional mutations as a progression of the cancer cells, heterogeneity and which cause the malignant neoplasia. So, this is a cancer pathophysiology, which is the series of the events. Now, regardless in the difference of the types, regarding the differences in the type of the cancer histologically and the physiologically, there is the existence of the common physiological process or malignant tumors or cancer development in the organism. Like the common accepted, the basis pathogenesis of cancer is damage to the genetic apparatus. Genetic apparatus cells like mutations, disturbance of the gene expression, activation of the tumor promoter genes or the inactivation of the tumor suppression genes. So, these are the some events. Now, it is believed that the genetic apparatus of the cell along with the inactivation of anti-tumor takes place, it's essential for the development of the tumor. Means inactivation of anti-tumor cells, which is the main cause of the malignant tumor, because there is no anti-tumor suppressor cells, they are just inactive. So, when they are inactive, the malignant tumors will arise. Now, but it should be noted that the inactivation of the tumor suppressor gene is one of the natural physiological reaction of the organism. When this reaction becomes pathological condition, the organism results the cancer development. Now, the mutation occurs in the various series. So, first is the mutation and the tumor initiations. So, here there is a genetic alteration which leads to the mutation in the single cell and which results in abnormal proliferation, that is known as the tumor cells, right. Second is a cell proliferation or the tumor progression. So, at that time the tumor progression continues additional with the mutation in the other cells which cause the tumor population. Now, these mutation cells have some selective advantage over the normal cells. They show the rapid growth, division, mutations and continuously they start the initiate to the tumor population. And now, third step is clonal section of the malignancy. So, clonal means they form the clones. So, the cell proliferation which leads a new clone of the tumor cells with increased rate of growth of other properties, and this is known as the clonal sections. So, clonal sections continues through the tumor development. So, tumor continuously becomes a rapid growth and increase the malignant. fourth step is the metastasis. So, metastasis is a complex process by which the cancer cells will break and they just invade from the primary region to the secondary region. So, they just, it is a complex region, primary circulate through the bloodstream, any kind of lymphatic system to the underside of the body. At the new site, the cells continue to multiply and eventually from the additional tumors comprised the cells get reflected to the original tissues. So, this is the metastatic cancer. Now, so these are the kinds of the these are the kinds of the series of the cancer, pathophysiology of cancer. Now, there are various therapies, there are various treatments of the cancer, just like for surgery to just remove out that particular area which is infected with the cancer cells. Chemotherapies, radiation therapy, hormonal therapy, targeted therapy, immunotherapy and laser therapy. These are various kind of therapies which are used for the treatment of the cancer. Now, first is surgery. So, primary method is used for the treatment of the most isolated solid cancers. So, they just diagnosed and they just isolate that particular area and they just surgically removed. Chemotherapy, so there are various kinds of drugs which are used for the administration of toxins to inhibit the cancer cells or they just kill the cancer cells. Now, radiotherapy. So, radiotherapy is a radiation that involves the use of radiations, ionizing radiation to specifically targeting the tumors within the body and leading to the death, and radiation is typically used in addition to surgery or chemotherapy for the certain types of cancer such as head, neck cancer and may be used alone. So, basically chemotherapies or radiations are the treatment which is commonly cause or commonly do for the treatment of the cancer. So, this is all about the pathogenesis, the pathophysiology of the cancer and the treatment of the cancer. The pathophysiology of cancer is, there is a series of the events in which basically the key point is the DNA repair genes get damaged. When the DNA repair genes get damaged, so there is a series of the damaged genes will form. When the series of the damaged genes will form, so what happened at that time? The mutation, that the mutated cell will grow and these mutated cell will cause the cancers. So, now at the end treatment, so there is a various kinds of a treatment which is used for the treatment of the cancer, just like radiotherapy, chemotherapy, immunotherapy and some kind of hormonal therapy. There is a various kinds of series. So, this is all about the pathogenesis of the cancer and the treatment of cancer. Thank you.